And now I'd like to introduce Dr. Vivek Sridhar, where he goes by he, him, and he is an assistant professor in the Department of Biology at UW and an affiliate researcher at the Max Planck Institute of Animal Behavior. His research discovers principles of spatial temporal computation that span across multiple scales of biological organization. To this end, he combines mathematical modeling, automated tracking, reconstruction of sensory information, and immersive virtual reality (VR) experiments with organisms from insects to humans. Vivek is also interested in communicating science with the public using various artistic media. As a mixed media artist and an alumnus of a field trip residency in the Academia Schulz Institute at Stuttgart, he collaborated with sound artists, writers, and filmmakers to foster curiosity and public interest in science and the natural field world. Vivek received his bachelor's in engineering in biotechnology from Manipal Institute of Technology in India. He then moved to Europe, where he received a dual master's degree, Master's of Science in Evolutionary Biology from the Uppsala University of Groningen. Vivek finished his PhD summa cum laude at the University of Konstanz and Max Planck Institute of Animal Behavior. He continued there for a postdoc and joined us here this summer. Everyone, I'm very happy to introduce him to you. Thank you for joining us today.
Thank you, Connor. Thank you for the introduction. Thank you all for coming here. It's a pleasure. I've just moved to Washington slightly over a month ago. I was in Germany, as Connor just said. So this is my first seminar since I started my academic appointment here. So it's great to be here and to talk to all of you. Actually, my first time in Seattle was also in SAS. I've done a small seminar at Andrew Burdell's lab. And so it's great to be here again. So I'm Vivek. I'm broadly interested in understanding animal movement, how and why animals move the way they do, how they search for resources, and how they choose among multiple such options, and the ecological and evolutionary consequences of these decisions. So I take a complex systems approach to the study of animal behavior. And in my lab, we combine theory with empirical work, both in the lab and in the field, to draw links across scales of biological organization. So thinking about how neurons within an individual's brain interact to produce individual behavior, and then scale up to think about how individuals themselves interact to produce collective behavior. In today's talk, I'm going to talk about two major directions of my work. One is thinking about animal decision-making, spatial decision-making specifically. And I will talk about some of the theory that we've developed along these directions, lab experiments that we conducted, and some ongoing work in the field. I will then talk about search, which specifically in the context of prey search in octopus fish hunting groups. So these are mixed species hunting groups that are looking for prey together. So these are the two directions I'm going to focus on for today's talk. All of the work I'm going to present today was done at the Max Planck Institute of Animal Behavior and the University of Konstanz. I'm also affiliated with the Indian Institute of Science, where some of my field work was made possible by in India. And I'd like to thank all my collaborators, my advisors, the technical and animal care staff at the University of Konstanz, without whose support, none of this would be possible. Whether you're an insect trying to find a tall blade of grass to perch upon, a fish deciding which amongst many conspecifics to school with, a primate trying to find a foraging patch, or an antelope trying to find mates. Most animals at some point in their lives make decisions on the go. But a lot of our understanding of decision-making comes from experiments like this. So on the top, you see what is called the dot motion paradigm. This is a common paradigm used in neuroscience to study decision-making. So the rat is shown dots moving on a screen, and it decides whether majority of the dots are moving to the left or the right. And if it presses the lever correctly and makes the right decision, it's given a juice reward. Otherwise, it gets nothing. In the field of animal behavior, we've used these binary choice assays. So here I've shown a schematic of a focal fish separated by glass partitions from two conspecifics, and we measured the animal's choice at the proportion of time it spends close to one conspecific or the other. And while we've learned a lot about how animals make decisions from experiments like these, these experiments miss a fundamental aspect of decision making, and that's one of space and movement. As a community, we've tended to think of decisions that's something that happens within an animal's brain, and for movement to be a mere readout of a decision that's already made. But today, I'm going to argue that movement is in fact a fundamental part of the decision-making process itself. And a lot of this starts from thinking about how animals represent space and options within their brain. So here, I'm going to start by talking to you about the fruit fly's brain. So this is the central complex, which is a specific region implicated in navigation within the fruit fly's brain. So here we have a circuit called the ring attractor. So we have neurons that encode different directions, and neurons that encode similar directions excite each other, and neurons that encode different directions inhibit each other. And what this means is that you get this activity bump. So here I'm going to show you a video, and that's actually video from the brain of a fruit fly. You see that activity bump, and what you will see is that it faithfully represents the location of the blue bar on the bottom left. And what this tells us is that options in space are actually represented as vectors within an animal's brain. And what that further means is that if options are encoded as vectors, when an animal moves through space, its internal representation of these options will change, and which in turn will feed back and affect its movement, which in turn will affect its representation of space. So there's a recursive feedback between an animal's movement and how it represents space and options. So what I did during my PhD was build a ring attractor model incorporating this recursive feedback between movement and an animal's internal representation of space to understand how animals choose among spatially distributed options. So the first thing I did was expose this model to a scenario with two identical options. The model predicted that the animal would spontaneously move in the average direction between the two options and abruptly switch to choose one option or the other. Here you see several hundred replicate simulations, but each time, of course, the animal will only go to one option or the other. And what this results is in a bifurcation. This bifurcation in physical space is also accompanied by abrupt transitions in neuronal dynamics within the animal's brain, or that's what our model predicts. But why do we care that the animals exhibit these bifurcations? Why do we care that these patterns sort of are predicted by these models? Well, we care because from statistical physics, we know that any bifurcation is accompanied by a peak in what is known as susceptibility. And this is a beautiful property for any decision-making system to have, because what this means is that the animal is extremely sensitive to small differences between quality of the two options. So if one option is slightly better than the other, the animal will almost always go to this option compared to the other. So this is a beautiful property for any decision-making system to have. So what we predicted is that going through this bifurcation, as you see in the left, is actually beneficial for the animal's decision-making accuracy. So here I've shown, again, trajectories. In both cases, the right option is better than the left one. The X is where the animal starts moving, and the two options are represented as blue circles. And in the left scenario, we see a geometry where the animal is allowed to pass through this bifurcation, whereas in the right, the options are diametrically opposed in a geometrical configuration where the animal is not allowed to pass through the bifurcation because it's beyond it already. And what our model predicts is that the animal should make better decisions when it's allowed to pass through the bifurcation. So we've long known that the time taken to make decisions is fundamental to an animal's decision-making accuracy. We've known that the difference in quality between these options is fundamental to decision-making. We think we've identified a third fundamental feature that affects decision accuracy, and that's one of space and geometry. Now, this is great. We have a beautiful theory. We expect certain patterns in animals' movement, and we have predictions or expectations as to why this should hold. But is this really true? And so to test this out, I conducted experiments in virtual reality with walking locusts and flying fruit flies. How does a virtual reality system for an animal work? Well, it works through what are known as anamorphic illusions. So here I'm showing you a Rubik's Cube that's placed on a table, and I'm telling you this is an illusion. But this illusion is so good that it fools you to thinking that this is a three-dimensional object placed on a table. But this illusion will now shortly be broken, and you realize that the reason it held was because of the perspective you were looking at it from. And this is exactly what we do in the VR, but instead of paper cutouts, we use high-speed projections, and so we can constantly correct for perspective from the animal's position, thus creating the illusion of a 3D world around it. And indeed, as predicted by our theory, both flies and locusts exhibit this bifurcation. They both move in the average direction and then choose one option or the other. But in the wild, animals typically are not restricted to choosing amongst two options. Most models of decision-making consider binary choice, but what about more than two options? We exposed our model to a three-choice condition, and our model predicted that the animal should move in the average direction between the three options. When the two extreme options exceed a certain angular conflict, the animal eliminates one of these options, going in the average direction between the two remaining options. It then does a second round of elimination to go to the only option remaining. And in sequentially eliminating these options, what the animal has done is it's reduced the complexity of its environment. It's reduced a three-choice decision to two binary decisions. And this is what both the flies and the locusts do. They reduce a multi-choice decision to sequential binary decisions. Our model is able to do this for any number of options. And here I have videos for radially symmetric scenarios up to 8 options. Now, thinking about the functional benefit of this, I mentioned to you that going through this bifurcation should increase the animal's decision-making accuracy. And working with PhD student August Paula and master student Lisa Ecker, we tested this out. So here we presented locusts with two options. The black target is the better option than the gray target. And you see that when the options are presented 180 degrees apart, the animal is only doing slightly better with the black option than the gray option. But when the options are presented at about 50 degrees, it actually amplifies the differences between these options, and the animal makes much more effective decisions. Now, if this is a fundamental property of how animals make decisions, then it should be widespread across taxa. I've now spoken to you about flies and locusts, which are invertebrates, and we'll now move to talk about vertebrates, and we'll talk about larval zebrafish. And here, once again, we presented the larval zebrafish, two virtual conspecifics that move back and forth parallel to each other in VR. And as predicted by our theory, the zebrafish, the real fish, would move in between the two virtual fish when these fish were close to each other, but as you increase the lateral distance, the real fish chose to swim with one fish or the other. When presented with three virtual conspecifics, we again see this breakdown of this three-choice decision to sequential binary decisions. Now, the patterns here look very different from what I showed you earlier, but this is the exact same model with the exact same parameters. The only reason the patterns look different is because of the moving frame of reference from which I plotted this. But otherwise, it's exactly the same. So far, everything I've shown you has been in bilaterally symmetric scenarios. What happens when we break this? So we presented the fish with three virtual conspecifics, two on one side and one on the other. Most models of decision making would predict that the real fish should swim twice as long, or more in any case on the right with two fish compared to the left where there's a single fish. But our model predicted that the real fish should swim roughly the same amount of time on either side. And this is in fact exactly what the real fish did, and we were ourselves surprised at the quantitative match at where the peaks turned out to be. So what we've shown is that this algorithm of decision making, we've identified it, we've focused on the benefit of it, and we've shown it to be true both in invertebrate and vertebrate systems, and both in where the options are static environmental targets, but also when the options are social. So what does this tell us about schools of fish when they're all moving together? So what I did was then I basically went to schooling fish data. So we have schools of 10 and 30 fish. These are golden shiner fish. And I analyzed movement of these data. We analyzed leadership and social influence in these groups, thinking about which other fish are you following and what are the factors that affect you following a certain individual. And what popped out as being most relevant was a vectorial representation of the neighbors. So the distance to the neighbors and the direction where this neighbor is. So agreeing with our initial, where we started all of this, that a vector-based representation of these options is what's driving these decisions. And we also now have ongoing work where we're starting to find similar patterns with humans. This is work by Francois Forster, who's running these experiments with humans in virtual reality systems. I'm now going to take this further to think about spatial decision-making in ecologically and evolutionarily relevant context. And this is in the context of legs. So this was work with Akanksha Rathod and Himal Nayak. They're two postdocs, the three of us collaborated on this work. And so we wanted to understand how female mate choice works on legs. So this is an iconic North American lekking species. So you see the sage grouse here. The males establish cluster territories and the females choose mates through these within the lek. And this is a great system to study spatial decision-making because the males provide no benefit to the female except for mating. They don't provide parental care. There's no resources in the territory. So this is fundamentally a spatial decision-making problem. And it's nice because the males are very site-fidel. They're very, they don't move very far from their territories. So we have a spatial geometry that just comes from how these males establish their territories. And so we set out to ask, what are the spatial and social factors that govern mate choice and mating success on a lek? And so this is the system which we studied. I showed you the sage grouse as an example, but this is what we studied. These are black buck, these are antelopes in India. The black ones are the males, the brown ones are the females. And what we did was we used drones to record videos of these animals. So I'm going to play a video, but effectively, this is what you'll see. Each of these circles is basically the male's territorial markings. So it pees and poops and marks its territory. And again, the darker individuals you see are the males and the lighter individuals are the females. And so what we do is like we record these videos, aerial videos of these animals. And because each drone's battery life is only about 15 to 20 minutes, we use relays to record longer duration. So we record about 2 hours, 2-hour sessions. And then we use state-of-the-art computer vision algorithms to track these animals. And so now we have their identities over the course of our recording. We've also reconstructed the landscape. So this is actually not a video, this is a 3D reconstruction of the leg. And so we now have a 3D map of the leg with latitude and longitude coordinates of each point on this on the leg. And so we have maps like this generated of the entire area just to give you a sense of the scale. This is about 120 meters, so the entire leg is about 300 by 300 meters. The breeding season lasts about 3 weeks, and we do 2 recordings each day, one in the morning, one in the evening, and about 2 hours of recording in each session. So that's kind of given us more than 3,000 videos with about 150 drone hours of footage. And we've sort of tracked all these videos like I showed you within their respective frames. But of course, the drone itself moves. So tracking the animals within the drone's frame is not good enough to actually measure interactions and get something about their physical movement. Of course, we have this over multiple years. So what we do is that we have these tracking videos, we have our map of the entire space, and we can use these territorial constellations to then georeference these trajectories. And then we can move from pixel coordinates to georeference space. So now we have latitude and longitude of these animals. And as you'll see, this is only a small portion of the leg, but of course the leg is much larger. So we do this simultaneously with 3 drones to cover as much of the leg as possible. And so this is the type of data that we have now. We've registered them. So this is just a sample of trajectories. You can see clearly when animals move from one drone to the other. And we have now trajectories of these animals with on average about 60 centimeter error, which is much better than using GPS collars on these animals, which would take us to several meters. So we have much higher resolutions tracking, and we can get at their behaviors much better because we have a video along with our movement data. And so this is where this work is at. So this is ongoing work. And that's sort of the decision-making side of the work. But as you'll see, everything that I've spoken to you so far, there's a fundamental underlying assumption that the animals already have information about all these options that exist in space. But animals don't always know where food or mates or social partners are, and they sometimes need to search. And this is where I'm going to jump to the next part of my talk, thinking about how these mixed species groups of octopus and fish hunt together, how they search for prey together. And here we'll talk about how leadership and social influence in these groups come together. This is collaboration with the marine biologist Eduardo Sampaio. Eduardo led this work. I was more involved in the data analysis and interpretation of the results. And so what I will tell you now is how leadership within these groups is multidimensional. Andrew last year published a paper talking about how temporal decisions are the next frontier of collective intelligence. So here we're going to talk a little bit, we're going to talk about how decisions within these groups actually can be split into spatial and temporal decisions, and how we can sort of think about these different aspects. And I'll show you that different individuals actually come out as leaders in different contexts. This is the type of data that we have. So we have videos of these octopus hunting with a bunch of these fish. The octopus is what is known as a speculative hunter. So it probes cracks and crevices in the ground. The other fish are waiting around. If any prey is flushed out, they will eat it. And this was the traditional understanding. The traditional understanding of the system was the octopus is doing all the work, and the fish are only there to eat any prey that's flushed out, that the fish give no benefit to the octopus. But I'm going to sort of build the story to tell you that it's otherwise. So Eduardo used this stereo camera rig, and this is how he followed these animals underwater. So we have these two cameras, so we can track these animals in 3D. We can reconstruct the ocean floor and get their trajectories through this. And this is what we analyzed, and this is sort of roughly when I came into the project as well. And we analyzed these data through what's known as like pull anchor analysis. So we think of these social interactions between these animals as spring-like interactions. So this is basically analysis that was developed in this paper, Stanford Preshkin and Farin et al. And there they used similar analysis to think about social influence in baboon troops. And here basically we adapted it from 2D to 3D to think about mixed species groups in these octopus fish groups. So when I just said that we think about these social interactions as spring-like interactions. So you have these two individuals that are relatively close to each other, then they go far from each other. And this could be because one of them initiates a movement, so it moves further away. And then they come back together, and they could come back for one of two reasons. The first being that the initiator actually successfully managed to get the other individual to follow it, in which case we call it a successful pull. The initiator is the puller and the other individual is the follower. The other reason they could have come back together is because the initiator actually came back, and this is an unsuccessful pull or a successful anchor. And we call the individual that was behind the anchor, and we call the initiator, we say that the initiator was anchored. Now, if we want to think about leadership within these groups, of course, the first thing we could think about is that the initiator is the one deciding where the group will go. So whoever makes the most initiations is the leader. And if this is how we think about leadership within these groups, we find that the goatfish are actually by far lead much more than the octopus or any of the groupers. The octopus and the groupers don't initiate many attempts. Most of the movement attempts are initiated by the goatfish. But you're not a leader if you initiate attempts and nobody follows you. So we came up with other metrics called pulling and anchoring efficiency. So this is your how often were you successful at pulling or anchoring relative to the number of initiated attempts? So how much do others care about your decision? So how much weight do you have proportional to how much you initiate? And here we found the octopus comes out by far as the leader. The octopus has the highest pulling and anchoring efficiency. But once again, even if everyone follows you when you initiate, you're still not a leader if you never initiate. So the functional leadership within these groups has to balance these two, the number of initiations, but also how often are you followed when you initiate. And so we came up with a metric of functional influence here. And this is what we find. So what we find is that the goldfish has the highest pulling frequency. And what this means is that the goldfish is the one actually making most initiations and is being followed the most number of times. The octopus, on the other hand, has the highest anchoring influence. So the octopus is the one that keeps the group. So how we can think about this is that the goldfish actually moves out and provides options as to where the group could move. And the octopus is deciding when the group actually moves. So the goldfish is making spatial decisions and deciding where the group goes, but the octopus is making temporal decisions regarding if and when the group actually moves. And so this is kind of what I just said. How am I doing on time? Perfect. So great. So we know that the goldfish is making spatial decisions, the octopus is making temporal decisions. Now, but these, I've only shown you the full network assemblages. These are when all species are present within these groups. But of course, in a lot of these videos, we have a subset of the species as well. So we use these as natural experiments. So let me go back. Apart from the octopus, the next the strongest anchor, of course, was the black-tip grouper. So of course, since all our groups fundamentally have the octopus, that's how we start recording this. So we use the natural variation within our group composition to identify what are the roles of these pullers and these anchors. So we looked at groups where there was no blue goldfish and where there was no black-tip grouper. In the absence of the blue goldfish, what you find is that the other species increase their initiation attempts. Here, you find both the barbel goldfish and the octopus increasing their initiation attempts, but the black-tip grouper doesn't seem to pull its weight. It doesn't seem to change its behavior very much. So it seems like the group needs much more initiation in the absence of the primary initiator, but the black-tip grouper doesn't seem to pull its weight, but the other species seem to increase the initiation to keep the group functioning together. In the absence of the black-tip grouper, everybody's pulling efficiency seems to increase, especially the octopus. The octopus seems to gain a lot in the absence, a lot of influence in the absence of the black-tip grouper. And so what we find is that, and also, I didn't show you the figure, but what we also find is that the group also moves, is much more mobile and moves a lot more in the presence of the blue goldfish. So the blue goldfish is like an excitatory stimulus to the system. The presence of the blue goldfish allows the group to explore a lot more. Yes, and in its absence, all other species seem to increase their initiation attempts except the black-tip grouper. The black-tip grouper, on the other hand, acts as an inhibitor to this system. It keeps the group less mobile, it keeps it more stationary, which is consistent with the fact that it's actually an ambush predator. And in the absence of the black-tip grouper, the pulling efficiency of most species, especially the octopus, increases. So what we've seen now is that we have this group, we have species that have high pulling efficiency, that are high pullers, that are spatial decision makers. You have anchors that are much more temporal decision makers. And the black-tip grouper doesn't seem to pull its weight in this group. Even in the absence of the primary puller, all other species put in more work, but the black-tip grouper doesn't. And so this is where we come into partner regulation mechanisms. And so this is effectively aggression between individuals. So to fish, we find chases, and for the octopus, we're talking about punches. So I have this compilation video of octopus punching other fish for you. And so if we start thinking about these partner regulation mechanisms and who's actually regulating whom, we draw this network, and what we find is that nobody exhibits any aggression towards the octopus, and the black-tip grouper, which is the cheater, is the primary receiver of all the aggression from all the other species. We now specifically focus on the octopus, and we see that the octopus primarily punches the black-tip grouper, and the other species don't get punched that often. And in fact, in groups where there is no black-tip grouper, the octopus never punched any other fish. These other punches are also in groups where the black-tip grouper was present. And we see also that the probability of punching is much higher when the group is more stationary and less mobile. We then went on to ask, what are the functional benefits of being in these groups? We know from the literature already that the octopus gains from being in these, the fish gain from being in these groups because they eat prey that are flushed out by the octopus. But what does the octopus gain? So Eduardo set up baits underwater, so the baited experiments where there were structures that were empty and structures that were full. And on the y-axis, we have the web over duration. So the web over is the behavior that the octopus displays when it's feeding. And we see that the octopus does a web over for much longer in a full baited, in a bait that's full compared to empty. And then when we look at these groups, in full groups, in the absence of the black-tip grouper, in the absence of the blue goatfish and the octopus alone, we see that the web over duration is highest in a group with the blue goatfish and without the black-tip grouper. So what the octopus does is it does fewer web overs, and it's actually spending more time per single web over, suggesting that it's actually feeding much more in the presence of the blue goatfish, telling us that actually, compared to the black line, which is the octopus when it's alone, so telling us that the octopus is actually gaining from being part of these mixed species groups. So I will end my talk there. Basically, with this work, we show that leadership is multidimensional and species so role specializations here. So we have spatial and temporal decision makers. There are investment asymmetries within these groups, and that results in the emergence of partner control mechanisms exhibited by the octopus punches or fish chasing each other, and that hunting in these mixed species groups improves everybody's success, foraging success, including the octopus. Thank you.
Yeah, thank you. And yeah, we have some time for questions. And people in the Zoom chat, you can just type in questions if you have any. And then I think since we are recording.
No green box around the screen.
Oh,
still sharing this?
Yeah, I think we are.
Okay.
Yep, we press that are sharing. So then let's go chat. So yeah, Zoom chat, if you have questions, just type them in. And then for people that aren't here in the room, you could just like repeat questions. Yeah, of course, perfect.
Yeah.
I had a question about some of your work, the first kind of project you presented with the locusts. So in the first bifurcation plot, the locusts seem to always go to the left, but there was a lot more.
So, I mean, I think they're roughly going equal to both sides. It shows up that way on the heat map because the heat map is capturing spatial density, so they're a bit spread out on one of the arms. So it looks less, but they go roughly
so
equally to both arms.
Gotcha.
Yeah.
Okay. And then later when you had three choices, or sorry, no, with the fish, and you split them up unevenly. So there was one fish on the left, two on the right. Why did your theory predict that it would be equal?
So it comes down to the geometry. So if we'd had 1 and 2 and we'd had it in a different angular configuration, you would have gone much more to the 2. But basically, because what these ring attractors are doing, they're integrating vectors in space. So like basically, if you have two options that are roughly pointing in a similar direction, the neurons firing for that direction are already saturated. So basically, you don't add up if you're like have several within the same small angle. Basically, if I kind of changed the geometry where I had two options that were more spatially spread apart, then the animal would have gone to that direction much more.
Gotcha, so it would have registered it as like more fish there
Yeah.
versus with the angle, it was almost just like still one fish.
Yeah,
there.
it's like a very small angle on that side.
Yeah.
Yeah. That's what we think. I mean, of course, we don't know if that's how it plays out in the brain.
Mhm.
Based on our understanding of the model and interpreting the behavior, that's what we think is happening. Yeah.
This might be a similar question to yours. And it was kind of like when you had three options, like they wouldn't just go straight for the center option. They would angle either the bottom or the top and then angle there. Is that sort of the same reason, or why is that?
So that's the bifurcation dynamic. So basically you have three options, and because what you're doing is when options are close to each other, they're activating each other. When they're far apart, they're inhibiting each other. So invariably the two outermost options are going to inhibit each other before anything happens to the middle option.
Mhm.
And that means that one of them is going to outcompete the other, and then you start going in between the remaining two options.
Mhm.
Yeah.
Mhm.
Yeah.
Has anybody been able to directly measure neuronal activity when they're like faced with bifurcations like this?
So I mean, I think, so I'm not involved. I know Ian is talking to my PhD advisor with whom I work. He's talking to neurobiologists to record neural activity simultaneously, but not me. Yes.
Really great talk. I was thinking about in your liking example, I was thinking about other mating systems. Do you guys have any plans to examine that kind of work in, say, like a heron system where males are controlling multiple females?
Not yet. I mean, haven't considered it. I think, yeah, I mean, I would be happy to chat if you have thoughts, and we could discuss it. Yeah, yeah.
Yeah.
But I haven't considered other systems yet. Yeah.
I was excited by the octopus punches naturally. So I was wondering, like, what do you think the octopus is perceiving when it's making these punches? Is it visually that the fish it's targeting isn't moving that much? Or does it have some abstract notion of who is working and who's not?
I mean, I would say it's I mean, this is all speculative, like you cannot know what the octopus is perceiving, but I mean, the fact that this group is keeping this fish is keeping the group from moving means that they're not foraging. Basically, they're not encountering more forage rate. So it could be as simple as when you don't forage as much, you give out a punch, and the black tip tends to be, because it moves less, it also tends to be closer to the center of the group near the octopus, whereas the goldfish are out further because they're exploring much more.
I guess I have a question which is like a continuation. So why is these anchor fish still part of the group? Like why they don't?
Yeah, I mean, it's a good question, but I mean, I think it's also, I don't know how much control they have on deciding whether this fish follows them where they go or not. Like basically, the octopus is it's not that they get together and then they start foraging. So they start foraging and then fish will join. Sometimes there will be fish that leave the group and other fish that come in. So it could just be that the blacktip grouper joins at times and then.
But they are deciding to let that fish anchor them.
Yes, that's true. That's true. Yeah, I don't know. Yeah, that's a good point. Yeah. I would have to talk to Eduardo for this. I'm not the marine biologist. I think I also don't understand the aspects too well. Yeah.
You looked at the octopus foraging efficiency, but did you look at the foraging efficiency for the other fish, like were they?
We did, but there is prior literature saying that the other fish gain from the interactions. So we focused on that simply because it's super hard to see when they're feeding. With the octopus, we could set up these baits, and the web over gave us a proxy of feeding. Yeah.
In the ring attractor model where you resolve bifurcations, there's got to be some stochasticity in that model. And I'm wondering how you ground truth the noise that you're putting into the model. It seems like that would influence when the bifurcation.
Yes. I mean, the noise definitely. I mean, so in the model, for example, that's why we kind of we're not making, like, the model here is more qualitative, we're not making quantitative predictions necessarily. But invariably, so basically, I don't have it here, but there's not, so if you look at the phase diagram, there is a critical noise level. Below that, the bifurcation will always occur. The angle itself might change quantitatively a bit, but it will always occur. Above that, you get into really high noise regimes where they're almost doing random walks. They go to 180 degrees and almost doing a random walk left or right. So we're not in that regime. As soon as we're below that, we're always seeing a bifurcation. The angle may vary, but
And the prediction is the bifurcation, not the exact angle or place where. the
Yes.
bifurcates.
yes. And also, that is not the prediction, but that it is curious that the fish, the locusts, the flies, even the humans data that we had is preliminary. The bifurcation, there's work in on baboons in the literature. The bifurcation angles are not, I mean, it depends on what you mean by hugely different, but they always occur between 70 and 90 degrees, kind of. And so there seems to be some level of consistency in wherever we are finding it. But I think so in the model that kind of comes from also from your nonlinear perception of space. So basically, if you within this ring attractor, if you say your perception of space is Euclidean, so interactions between these neurons is perfectly Euclidean, like cosine based interactions, then these bifurcations will happen over at 120 degrees or super high. So we know that's not the case. So there is a non-Euclidean perception of space, which actually gives us bifurcations at this level. The type of bifurcations we see, it's an elliptical geometry rather than, Euclidean geometry. But that's, I mean, we're not making predictions on exactly what angles.
Yeah, this is like, it's super interesting because I feel like it gives you a glimpse into like what the personality of like these fish could look like. And I think it's really curious that like, well, say you take a fish out of a system, then like the behavior of all these animals completely changes. And I think what I'm curious about is like, and when fishes and populations are being driven to extinction from like maybe say fisheries overfishing, like then you're gonna lose these species, and then like that behavior, like I guess in relation to this theory, like with these results would kind of change. And I wonder like how, like how do you understand maybe like across other species, like is this something that could be used in like a management context, or I guess how does that play into understanding? Because it's certainly, because I would think there's animals that may not like have or be suitable for that different personality, and thus like they don't adapt and then they fall off.
Yeah, no, it's absolutely, I mean, it's a good point. I mean, I've never talked so much about the management implications. I mean, I think it's a little bit like, like I've been thinking about the functional role of the species within the network.
Mhm.
I mean, I think you can use some of that, but there's also problems in making management decisions based on specifically what perceived role you think it plays within that ecosystem. I mean, I think so. Eduardo also went and looked at octopus. So a lot of these data were in the Red Sea, but he also went and looked at the same octopus species as in Japan as well. But there it swims with a different assemblage of fish. We didn't analyze those data, but his intuition spending time with them was that there are other species there that they seem to interact at least visually similarly, but so there are other species that seem to play similar roles, but
Oh, okay. Cool.
I'm wondering, we were around about this, but like any configuration of individuals, if they're having this like ring attractor model, thinking about their conspecifics as like potential targets of interest, like any spatial configuration would give a different susceptibility level to each fish. And so, there's sort of these characteristic spacings and arrangements that fish move in groups. Do you think that those arrangements that emerge are ones that like peak the susceptibility? related to that.
Yeah, that's interesting. I mean, I think I haven't, it would be interesting, like, I think with the schools of fish that I looked, I said, you're following ones that are a vector-based representation of the neighbor. But also if you look, the angles where you start or stop following these individuals also fall similarly within the 70 to 90 degree range. So you're not following someone right there, but you're following someone about there. So kind of if you take those angles, they also seem to fall similarly, but I'm also very wary about making any quantitative claims or predictions with that. There's one of Ian's postdocs, Mohammed, who kind of then expanded some of this work to build a new schooling model where each agent has a cognitive model rather than a certain particle, and you do get some schooling behaviors. I think probably still a way before. Yeah.
Any,
Yes.
Is there like a gradient version of the ring attractor? So instead of
Yeah.
discrete particles?
Yeah, so we were working, like I had a student with whom I was working to do this in a gradient, like starting with a 1D gradient and kind of, we didn't get very far, but I think, we started working on it, we had some thoughts and directions, but didn't get further than that. Yeah, but I think it naturally gets there. Because in the end, this ring, you're giving input now with discrete, we're just giving it Gaussian inputs. And instead of Gaussian inputs, you need to figure out how you want to add up this different 1D Gaussians and then create a gradient, and it should function. And the nice thing about the ring attractor is that you will always only ever have a single bump, which then tells you where you integrate all these vectors and where you end up wanting to go.
Yeah, yeah. I wonder with like eyes or nostrils or ears that are sort of like orthogonal to our direction, their separation is orthogonal to our direction of travel. So if you start going up the gradient, then they start to measure equal. And so then they're like, there's less information about which way to go when you're going the right way.
Yeah,
or the wrong way.
yeah.
any other